tes.
Recording in progress.
Good morning to everyone that's already in the room. My name is Shinta, and I will be your host for today's guest lecture. I'm the student of Master's Program in Biomedical Science from Department of Parasitology. And today I would like to welcome everyone, Professor Wabai, Dr. Ada, and Honorable Lecturer from Faculty of Medicine, Universitas Indonesia, also the lecturer from South Korea. Welcome to Faculty of Medicine, Universitas Indonesia. And at first when I saw the poster, I was very intrigued because malaria in South Korea, I hope that you are as intrigued as I am and excited to know more. And of course, that also being said, what have we done as Indonesian, especially in Department of Parasitology, to overcome this disease? As we all know, it's a tropical disease, and how far and wide our effort to combat such disease. Yeah, we're going also to learn the lecture from both sides, both from South Korea and from Indonesia about this particular disease. And this event will not be held without a beautiful cooperation between the master's and doctoral program in biomedical science from Faculty of Medicine, Universitas Indonesia. This guest lecture will also be led and moderated by our own Dr. Robiatul Adawiyah, Spesialis Parasitologi Klinik. And before we delve into this guest lecture, I would like to read Dr. Robiatul Adawiyah's curriculum vitae. Robiatul Adawiyah, MD, PhD. Her current focus is developing diagnostic method for systemic mycosis, including histoplasmosis. She is part of the team development of antigen for serologic testing and molecular epidemiology study on strain of Indonesian histoplasmosis capsulatum. She has been accustomed in collaborating in research with Retno Wahyuningsih since 2008 and also with Sam Samuel Surya since 2017. She's the Vice President of Collegium of the Indonesian Society for Medical Specialists in Clinical Parasitology, the General Secretary of Indonesian Society for Human and Animal Mycology, or INSHAM, and also the member of the International Society for Human and Animal Mycology, ISHAM. Based on that, she has been accustomed to organizing national and international scientific activities. And I would like to also acknowledge for the audience that follow this guest lecture online. Welcome to the event. And now, please welcome everyone, our own Dr. Ada.
Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you, Miss Sinta. I will invite for a guest lecture. The first, we will hear the lecture from Dr. Jin Hee Han from Kangwon National University College of Medicine. His topic is Trends in Tropical Medicine in South Korea and Malaria Research. For the first, I will read his CV. He got a bachelor from Kangwon University. He works in Department of Medical Environmental Biology and Tropical Medicine, School of Medicine, Kangwon National University, Chungcheon. Is that right? Chungcheon, yeah. Uh, Chungcheon, okay, Republic of Korea. He got a bachelor from Kangwon National University. He got a MSc and a PhD from Kangwon National University, and then postdoc from University of Otago, New Zealand, and also the last assistant professor from Kangwon National University. Please be my guest. Sorry. Jin Hee Han, Dr. Jin Hee Han. Okay, time is yours. Please, welcome. You have 20 minutes. Okay.
Okay. Thank you so much for inviting me. Yeah. So how can I see my presentation in here? Okay, okay. Yeah, I'm Jinny Han from Kangwon National University. So today I'm gonna talk about the trends in tropical medicine in South Korea and how about the malaria research. Thank you. Okay, so today I'm going to talk about the South Korea, not the North Korea, because I don't know the situation about the North Korea, but because maybe the malaria situation may be 10 times higher than South Korea. Just we guess like that. Yes, but this is not exact number. We don't know actually, but just in South Korea guess like that. Okay. Before start, I just introduce by myself again. Yes, I'm starting from the PhD course in 2013 from the Kangwon National University, and my major topic is Plasmodium vivax. This is one of the malaria parasite. So my department is the parasitology, and 5 years later, I got the PhD, and luckily I moved to New Zealand, especially in University of Otago. At the moment, I'm focusing on the Plasmodium cynomolgi. This is a zoonotic malaria parasite. So naturally, they infect the monkey, not the human, but recently we found in the human also. So now we call this is a zoonotic malaria. And 2 years later, also luckily, I got the permanent position in Kangwon National University again. So I'm back to Kangwon National University, and from the 2020, I'm starting to work. And recently, my major topic is the zoonotic malaria also. And this is me, probably 5 or 6 years ago. I look very young, right? Not like this. So, yeah, if you want, you can really contact me. This is my contact. So if you need, you just take a picture, and later you can contact me freely, or just ask me. Anything is okay. Okay. Okay, let's start talk from here. So we have to know what is a tropical medicine, right? So you know where is the tropical area? It's red color is the tropical area, right? So the tropical medicine is studying about the tropical area disease. Okay, the tropical area is, we can say this tropical area is abundant of the species, the live species. That means even the pathogen species, the number of species is very high, because they just like to survive in here because of the high humidity and high temperature. That means also we can say this in this area have a high risk for the infectious disease. So the kind of infectious disease, it contains the virus, bacteria, Rickettsia fungi, and also protozoa, including malaria parasite, and Hermes, one of the parasite. But actually, the tropical medicine is not only cover this infectious disease, also contain the non-infectious disease, like nutritional deficiency or anemia, poisoning, animal and plant toxicity, such kind of things. So in Korea is here, we are not in the tropical area, but why we need to focus on the tropical medicine, right? So there is the reason. So let's see the first, the trend in tropical medicine. In Korea, also study about or research about the tropical medicine. So recent trend is like that. There are three keywords. The first one is climate change. And second term is One Health. And third term is the international collaboration. So let's see one by one what this means. So climate change. Based on the global warming, the Korea, the tropical area now, the expanding in the northern part and southern part also. So in the future, even in Korea, will be change the climate to the tropical area. That means the infection disease or infected pathogen species will be increased a lot. So Korea government also want to protect from now in the near future risk. So because of that global warming, the expanding the tropical area will lead to the geographically expanding and divergence of pathogens. So in the future, even in Korea, it's not safe, right? So increasing, the result is like that, increasing the possibility of emerging infectious disease appearing. That is why even the Korea also focus on the tropical medicine now. So this is a typical example. This is one of the malaria risk modeling study from the Venezuela. At the past, there is the red color is the risk, high risk of the malaria infection. So there is a very small land have a risk area. But recently it increased because of the global warming. And in the future, they will increase more. But this is not only the Venezuela situation. Also, we can think about the South Korea or other subtropical area will be changed like this. Okay, so related to the climate change, there is many kinds of pathogen or non-pathogens studied a lot. So the best one is malaria actually, and dengue and other virus, other pathogens they studied a lot in now. So the second term is One Health in the tropical medicine. So what is the One Health? This is a very simple concept and important concept because previously just people thinking about the one pathogen and human interaction. Why they infect the human? How we can eliminate them? We just thought like that. But after COVID-19, the way of thinking has changed. So we have a very important question, where they came from, right? We have to cutting their connection to protect the human before got the infection. So there is many virus origins. So SARS virus also it affect in South Korea in 2002. They originate from the bat. They transmitted CBIT, and CBIT transmitted the human population. It's mine, no, right? Yeah. And MERS, this virus are the same. This MERS virus came from the bat, and they transmitted the camel, and camel transmitted the human populations. And COVID-19 also same. Just we guess now, the COVID-19, this virus came from the bat also, and they transmitted the pangolin, and pangolin transmitted the human. So if we can cutting this, any connection of this, the human, we don't need to get any viruses infection. That is why this is a One Health concept. So we have to understand the animal and human and environment. So to understand the one pathogen, we are not only focused on the pathogen, also our surrounding environment also need to understand. This is the concept of the One Health. So recently the Korean government also focusing on this whole map, how they connect to each other, something like this. So this is the same thing. So the people cannot live alone. We have to interact with some animal and environment also, right? That is why for the One Health, we have to think about this connection, animal and environment, even within the human population also. So to understand the one pathogen, we have to now use this concept, One Health. Okay? So even in malaria, it's the same, because the malaria parasite, we thought that the Plasmodium species is very restricted to select the host. But actually, recently we found a lot of monkey species in human, right? So to to reduce this risk, we have to understand the monkey behavior and the environment nearby. So this is One Health concept. Okay, the international collaboration is collaboration, just like this. So I move to the topic, to the malaria research in South Korea. So, you know, even in South Korea, have a Plasmodium vivax cases every year. We want to eliminate them, but it's really, really difficult. So a few years ago, they reduced around 500, and just during the COVID-19, they reduced to 200 cases per annually, something like this. But nowadays, increase again. It's really, really difficult to control the malaria, even in South Korea. Because the Korea, they have a borderline with North Korea, right? Almost malaria patient is appear in the nearby the DMZ. The DMZ is the demilitarized zone. Okay, so nobody can enter this area. Even the soldier also cannot be enter. Even they enter, they have to have no any weapon. Okay, that means we cannot control mosquito in this area. Nobody can enter here. That is why every year the malaria patients starting from the nearby the DMZ zone. So actually the South Korea government to say this malaria came from the North Korea. Okay? But North Korea also say this malaria patient came from the South Korea, something like this. We don't know actually what happening is here. And also based on the global warming, the malaria, the occurring area will be increased now. So even this year, the space, in the space, even the southern part of Seoul also have a patient. It's unusual actually, probably because of the global warming. Nowadays, this is a summer season in Korea. It's super hot and super humid now. It's more than Indonesia. Maybe you cannot guess. Korea is more hot and more humidity is more high. And the winter season is very cold. All freeze. Okay? So in South Korea, actually there is 3 university or institution working on the malaria research. One is the My Lab in Gangwon National University. It's nearby the Seoul. One institute, this institute is Institute Pasteur Korea. They also working on the malaria research. And in the southern part, there is another national university called Gyeongsang National University. They also focusing on the malaria research. So in Korea, only 3 teams focusing on the malaria research. So for me, I working on the protein characterization. So my research topic, usually I use protein science and protein-protein interaction, like that. And we made a lot of collaboration with internationally. Even me also, I made a collaboration with Indonesia and in Africa, Tanzania and India and China, and so I actually has a lot. And the Institute Pasteur in Korea, they specialize by this drug screening. They have a high throughput screening system. So as I know, only few institutes have this drug screening system. And collaborate with me, and they do the protein series structure analysis. And they also have an international collaboration with France and Cambodia, and there has a lot actually. And Gyeongsang National University in southern part, they focusing on the gene analysis. They are specialized about this, and they also make a lot of collaboration in Asian country. Like this. So in South Korea, malaria research led by this 3 group actually. Okay. So I'm talking about my research topic little bit more. So I'm focusing on the invasion biology. Okay. So this is a merozoite. So this tiny metazoite can express 6,000 to 8,000 proteins. Among the, some of the proteins are involved in the host cell invasion, we call the ligand. So they interact with some of the receptor in the host cell. So today, I'm gonna talk about this DBP and DRC interactions. So this is my ongoing project, actually almost done. So just open in here. So this is metazoite, right? And this red color we called micronin, the small organism, micronin. So in here, the DBP expressed. So this is the whole structure of a DBP, and this DBL domain, especially they are bind with the DRC. So this is a DARC, it's a human red blood cell receptor. They can interact each other. And if we block this interaction by using some monoclonal antibody, something like that, this metazoite cannot be invaded. So that is a very nice vaccine target, right? We can protect the invasion. So many scientists focus on about this. So this is a result of the in vitro. So if we use this 2C3 monoclonal antibody, this one against this domain of the DRC, this domain is this alpha helix domain. So if bind with here with some of the antibody, they cannot invade, they just die, right? So this is a very nice vaccine target actually, but many people are trying to develop this molecule as a vaccine, but failed because of the natural polymorphism is too high. So every people have a different structure in here, different structure of the antibody. So we cannot make a common vaccine. So we have to make individually fit vaccine. But this is really difficult to make like that. So anyway, nowadays, we cannot use this DBP-based vaccine. So we just change the thinking. Just screening the small molecule. The small molecule is like chloroquine or artemisinin and primacin. All of this is a malaria drug, right? So we designed the study as can a small molecule interrupt the PV DBP and DRC interaction or not? We want to find any molecule to interrupt this interaction. So if we can find, we can use experimental positive control because still have no control. Because this DBP and DRC interaction is the most important, most essential for metazoite invasion. But we only have a blocking agent in the DRC. DRC is a human receptor, so if we block in the human receptor, we don't know what is the side effect, right? Normal person, not the patient, normal person also have this DRC molecule, and this DRC also have their own role. So we cannot block that one, right? So if we are lucky, maybe in the future, we can develop the new treatment agent from this compound. So we can make this compound derivate also for induce more stable structure and induce more active structure. So we're starting from here. We collaborate with many of the institutes. So this is Korea Chemical Bank. They do the computer-aided drug design. It's a kind of virtual screening method. So we integrate with some of the advanced technique. So they're starting to screening 580,000 of the compounds. Especially this compound can bind with the DBP molecule or not, because we want to targeting the DBP. So they choose the 6,000 compounds. This one highly possible to interact with the DBP. And we're screening again in our university pharmacology department, using this pharmacophore model. And we select the 2,000 highly possible to interact with the DBP and interrupt DBP and DRC interactions. We're doing the in vitro study. We call it FPA. By this one, we choose the 26 compound. This is final compound. And within this, only 3 compounds are commercially available, so we can buy from the company. So we buy this 3 one, and we tested it in the ex vivo study. Actually, it's like ex vivo study. We use Plasmodium knowledge, one of the zoonotic malaria, right? But we already gene edited this one. So PK parasite, but they are expressed PVDBP, not the PKDBP. So they use PVDBP and human DRC interaction for invasion. So we use this one as a model. So this is some of the results. We screened 2,000 of the compound using the this fluorescence polarizing assay. We found 26 hit. It's more strong. The binding affinity is stronger than DRC, right? So if they are compete each other, this compound is more strongly bind with the DBP than DRC. So it can be highly possible to block the DBP and DRC interactions. So this 3 star, 3, 1, 2, 3, this 3 compound is commercially available compound, so we buy from the company. We tested in the PK parasite, but this PK parasite, knowledge parasite, express Plasmodium vivax DBP, right? This is using the CRISPR-Cas9 technique. We change the gene to express PVDBP. So the IC50 value is not too high actually, but anyway, they can block the merozoite invasion. So if we make, based on these compounds, if we make a derivate, maybe we can increase the more higher activity. And the ampicillin is famous, the antibiotics. They are not block the merozoite invasion. This is just negative control. Okay, after we move to the computational science again, and we check each compound, how they can interact with the DBP molecule. So this pink color is a DBP molecule, protein. And in here, this part, the DRC binding site. And this green color is our target compound. So we make a simulation how they docking with the DBP. We simulate 20 times. Among that, this compound is 60% docking in here, and 40% docking in here, and then also 60% docking in here. And the DBP binding affinity is also quite high with our compounds. So these compounds are also highly possible to block the DBP and DRG interaction. So in here, the yellow color alpha helix structure, actually here is the DRC binding site. So our compounds tightly bind with here, so they can block the DRC binding, like this. So now we are trying to integrate with the advanced technique like computational science and even the high throughput screening system for the research. Okay. So this is the end of my presentation page. So for this work, many people help us in even the pharmacology department, Professor John and Professor Lee and their student. And this is my team. There is 2 of the Indonesian students. They came from the Undip, actually. Yeah. And the Korea Chemical Bank and Institute Pasteur Korea and London School. From London School of the Lower to Moon also help us to make the parasite line. So the malaria research, not only malaria research, many kinds of research work cannot be done alone. So always want to make a collaboration and share the knowledge and help each other, something like this. So I have a lot of group now working with me together. So also here as Indonesia UI, right? So last time I put in here, UI is here. Okay, so yeah, this is the end of my talk. So just any question? Question is next time, right? Okay, so I want to say, just open your eyes and see the world, not only national. Okay, every research is need to collaborate each other. Okay. You have to be a global leader. You can do that. Okay. Thank you.
Test, test. Okay, thank you very much, Prof. Ji Hin Han, for the very interesting lecture. And then we will move on to the second lecture. The speaker is our lecturer here, Mr. Nur Hadi Eko Firmansyah, PhD. He is the lecturer in Parasitology Department, Faculty of Medicine, Universitas Indonesia. Okay, here is his curriculum vitae. He got a BSc in Jenderal Sudirman University of Indonesia, and then his master from IPB University, and then his PhD in parasitology in Khon Kaen University, Thailand. Okay, Pak Firman, here's your time. Time is yours. 20 minutes. Thank you. Applause for Pak Firman.
Okay. Thank you, moderator, for your kind introduction. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. First of all, I would like to thank to all of you coming here to attend this guest lecture, and also I would like to thank to head of master program in biomedical sciences, Prof. Waimuli, and then also it's an honor to meet you, Dr. Han from Kangwon National University, Republic of South Korea, and I think I see a lot of students coming here, and I guess all of you as a BTS fan and like watching Korean drama. Okay, well, so this is, as you look at the screen, this is my topic for presentation today. Recent update of malaria research at Department of Parasitology, Faculty of Medicine, Universitas Indonesia. So this is not only malaria research at the Department of Parasitology, but also it can cover malaria topic research in Indonesia. Okay. Okay, this is the content for today. Yeah. I would like to talk about the malaria situation in Indonesia, and then malaria research group in Department of Parasitology, and then what we did in the last years or last 5 years ago, and then future collaboration, as said Dr. Han. Okay, so as we know, malaria is a disease was caused by Plasmodium parasites and transmitted by Anopheles mosquito. Malaria is a major health and developmental challenge for Indonesia. In 2022, there were over 440,000 confirmed cases of malaria and over 2,000 deaths. 74% of Indonesian districts have been certified malaria-free, as we can see in the screen, but progress on the remaining 164 high-risk districts has stalled over in the last 5 years. As we can see in the screen that this is the map of Indonesia. The red color show the high endemicity area, in the East Indonesia, Papua and West Papua province. And the yellow color represent meso-endemicity area. We can see in the Borneo province, Papua province, and also in Maluku and Nusa Tenggara province. And then for green color represent low endemicity area, spread in the Sumatra, Borneo, Sulawesi, and several districts in Java, Maluku, and also in Nusa Tenggara. So, as we know, the malaria is transmitted by Anopheles mosquito. As you know, malaria caused by 5 Plasmodium species. We found 5 species in Indonesia. Yeah, Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and then the last is Plasmodium knowlesi that we found in patient. But until now, we don't know the update of Plasmodium cynomolgi in the human. But the predominant species are Plasmodium falciparum and Plasmodium vivax. The mixed species infection are common. So the Indonesian government has targeted malaria elimination by 2030. And however, case numbers have remained stable or increased slightly over the last 5 years. So, okay, so this is the people that involved in malaria field at our Department of Parasitology, Faculty of Medicine. The first is Prof. Inge Sutanto. She research topic is drug administration on malaria patient and drug resistant detection on Plasmodium. And then the second is myself. I conduct research about the malaria vector, Anopheles mosquito, and then simple method to detect Plasmodium in the blood. And then the last is Dr. Amar Hashim. They study about the vaccine development in malaria. So, okay, let's move to the first topic about from Prof. Inge Sutanto. She study about the agreement between serological data on school children and the number of malaria cases in the remaining high burden villages of Indonesia. So the study to measure malaria transmission in eastern Indonesia, where reported cases have been recently declining. So the study was conducted in Malacca district, West Timor, Indonesia. As you can see in the screen that this is the map of Malacca district. The red color represents one village that high burden malaria or annual parasite index is more than 5. And then yellow color is annual parasite index from 2 to 4 index, and then green color is low annual parasite index. So this is the cross-sectional survey of elementary school children aged 5 to 14 years residing in 6 villages. So the first protocol, they collect blood from the subject, and then finger prick plasma samples were tested using a multiplex Luminex to measure IgG antibodies against a panel of 8 Plasmodium vivax antigens, such as merozoite surface protein, rhabdoid-associated membrane antigen, reticulocyte binding protein, and then erythrocyte binding protein. And then the data was analyzed. So this is the result of this study. So this table shows the predicted Plasmodium vivax exposure status by village and age group. As we can see in the village group, the highest exposure was on Lamea village, and then followed by Weseben village, and then Seserai village. And then for age group, the highest exposure was on age group 11 to 14 years old, followed by 8 to 10 years old. So 15 of 398 school children were seropositive for recent Plasmodium 5X exposure. So 87% of seropositive children were from one village, the one with the highest API, and one seropositive child was from a village with API of 1.3 and another from a village with an API of 0.0. So that means that even the Plasmodium 5X cases was declining, but we detect with the serological test, we found this result like this. And then the next study about the simple method to detect the Plasmodium falciparum in the blood. So this study was conducted by my team when I studied in Thailand, the comparison of 3 methods to extract Plasmodium falciparum DNA from whole blood and direct blood spot or DBS. So we compare the effectiveness of 3 DNA extraction method. The first is the commercial method, the GF1 blood DNA extraction kit, and then we develop our method. Yeah, this is the TE buffer and proteinase K method for cell lysis, and then another commercial method is denazole direct DN131 for a single reagent combined with the heating for extraction process. So for the protocol, as you can see in the screen, that this is the common method when we extract the sample from blood like this. So consists of blood lysis until the elution. Okay, this is the method from commercial kit. And then next is we developed by our lab, yeah, TE-PK method. Well, for the first is from sample whole blood. So this is the flow method, like this, add a sterile distilled water to whole blood in centrifuge tube and vortex, and then centrifuge, and then discard supernatant, and add TE buffer and proteinase K, and incubate first for the first time to activate proteinase K, and then incubate again to inactivate proteinase K, and centrifuge again, and supernatant was collected, and use this supernatant as a template for PCR. And then for DBS sample, it is similar with whole blood sample, but the difference is in the first step. As I said, they add sterile distilled water to centrifuge tube containing DBS, vortex to dissolve blood into sterile distilled water, and then discard filter paper. And then next method is similar with the whole blood sample. So how about the denazole direct? So this is the not many protocol for denazole direct, as we can see in the slide. It's just add this sample and then centrifuge and then add the denazole direct reagent and then incubate and centrifuge. And also in the process for DBS sample, similar with whole blood sample. And then the genomic DNA we use for PCR, conventional PCR reaction, and we use primer to target the 18S rRNA gene, and then visualize under UV light. And then we assess the DNA quality and quantity, estimated cost and time required. Yes. Okay, so DNA concentration and purity of the 3 DNA extraction method were measured using nanodrop, and then the estimated cost was based on the price on the chemical and disposable items, including pipette tips and microcentrifuge tube, and then the time required for each method to finish one extraction from whole blood and DBS was estimated according to the procedure used in this study. How about the result? So we can see in the table the quality of genomic DNA. Its method, the highest quantity was in DN131. Yeah. And then for quality was in the commercial kit, but for estimated time was quickest in the denazole region, DN1313, and estimated cost, cheapest one is in the TEPK method. And then for from DBS sample, yeah, for optimized system, the quickest was in DN131 from the denazole region, and then for quality was in commercial kit, for quantity was in denazole region, for estimated time required in the denazole region, and then the cheapest one was in the TEPK method. For the comparison, for each technique, for each method, if we compare in the from whole blood and DPS, there's significant different, but for quality is no significant different for each method. So, and then we assess for limit of detection the Plasmodium falciparum from whole blood from the 3 method. So, if we see in the figure A, we saw the DN13 method, so a limit detection of 0.012 parasite per milliliter, and then GF1 PD kit, so a limit detection is similar with DN131, and then the TEPK method show a limit detection 1.6 parasites per milliliter. And then from DPS, figure A, so the DN131 method, so a limit detection 1.6, and then GF1 PD kit, so a limit of detection only 8 parasite per milliliter, and then TEPK method, so limit detection 200 parasites per milliliter. So the results indicate that the denazole direct region stand out as optimal choice owing to its cost effectiveness, time efficiency, and then superior LOD in comparison to both GF1 BD kit and the TEPK method for extracting DNA from whole blood and from DPS sample. In other hand, the TEPK method could be a suitable, more economical alternative for whole blood extraction compared with use a commercial kit. This finding offers great promise for application of this approach in large-scale studies in resource-limited regions such as in Indonesia. So, and then we have ongoing plans. We develop Accelerate IDN 2030, addressing the challenges of diagnosis of human malaria towards elimination in Indonesia by 2030. This is to establish the molecular tools to detect the presence of PFH-RP deletion mutant in Indonesia, and also to establish molecular testing capacity to detect zoonotic malaria parasite in Indonesia. This is collaboration with University of Glasgow. So For the next update, it's come from Dr. Amar. He studied about the adeno-associated virus-based malaria booster vaccine following attenuated replication competent vaccinia virus LC16M8 priming. So this study to compare the efficacy of different AAV serotype, AAV1 and then AAV5 as malaria booster vaccines following priming with the attenuated replication competent vaccinia virus strain LC16M8, which harbor the fusion gene encoding both the pre-erythrotic stage protein, Plasmodium falciparum circumsporozoite, and the sexual stage protein in two dose heterolog prime boost immunization regimen. But I don't talk much about this study because I have no experience about this study. So for the study, we want to develop the transmission blocking activity of viral vector malaria vaccine, LC16M8 prime and AAV1 boost encoding fusion gene, and PV25 PVCSP in Indonesia using Anopheles maculatus and Anopheles aconitus. So the aim of this study to evaluate the transmission blocking efficacy of the LC16M8 prime and AAV1 boost vaccine encoding the fusion gene PF and PV using membrane feeding assay in the Anopheles mosquito species in Indonesia. So this is the overview of this study. So the first step, we want to develop in the Kanazawa University in Japan, consists of group 1 and group 2. This group 1 from mice and group 2 from monkey. Group 1, we inject the PF vaccine, and also group 2 from mice, we inject the PV vaccine. PV, it's mean that Plasmodium falciparum, this PV for Plasmodium vivax, and also in the monkey, we inject in the group 1 PF and PV vaccine. And then after that, we collect the IgG or sera from mice and from monkey, and then mix with the blood patient, normal blood, and then Anopheles aconitus and Anopheles maculatus feed this blood in the membrane feeding assay, and then we count and we check or assist day per day from day 10 to day 12 and day 20 and to day 21 to sporozoid check in the Anopheles maculatus and Anopheles aconitus from Indonesia. This is the further studies. Okay, so this is the last, the end of this my presentation. So malaria have a big challenge and big problem. Yeah, as you know, the Indonesia have many island and then specific factor in the island. So that means that we, as Dr. Han said, that we have to collaborate with each other to combat malaria. So the future collaboration, if we want to development of molecular diagnosis of malaria, is also challenges in Indonesia because we need taxonomies or we need microscopies. That means that microscopies now is gold standard to check malaria in Indonesia. So we need to innovate or we need alternative method to diagnose malaria. And then the second challenge is emergence in the zoonotic malaria from Plasmodium knowlesi, Plasmodium cynomolgi, from animal to human, as you said, from as Dr. Han said, this in context One Health, we can collaborate each other to solve this problem, to solve this challenge. So this is my end of my presentation. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much for Mr. Firman. It's very interesting lecturer. And then please welcome Dr. Han, Professor Han, and also Dr. Firman to come here. We will go to the question and answer session. Okay, I will open 2 sessions maybe for question and answer. Okay, please, who will give question? Okay, please tell your name and your institution, maybe your program, and then please say your question. Okay, please, Miss Nurul, yeah?
Yeah,
Yes.
Doctor. Thank you very much, Doctor, for the opportunity. My name is Nur Gustiatima. I am a master's student in Biomedical Sciences, University of Indonesia. Thank you very much for both of the lecturers for the wonderful presentations, and I would like to ask questions to Prof. Han. So the pathogens is carried by mosquitoes, and then it can invade to human, the malaria. How about, so are there any research in South Korea regarding the prevention or the treatment of the pathogens in animals?
Okay,
Thank you very
please,
much.
Prof. Han.
Yes, your question is treatment for animal, especially on malaria or other?
Yeah, especially in malaria, probably in mosquitoes.
For
Are there
mosquitoes.
any treatment or prevention? Not like a drug treatment, probably in South Korea for animals.
So you mean how to control the mosquito, right? Yes. Yes. Some area we use the insecticide, but some area is not. Yeah, that is all. Is no more than that actually. Yeah, some area just we use insecticide, but especially our Aedes species has they are increasing of the insecticide resistance. So that is a new problem in Korea. But for Anopheles, yeah, we still use that one.
Okay,
So are there any
Other
increasing-
question?
Yes.
Yeah,
so
please.
Are there any increasing population with dengue, probably in
Population
South
with
Korea?
dengue. Oh,
Yeah, fortunately in Korea has no dengue virus.
Oh,
Yes, but we just have an imported case.
oh, important
Ah.
case. Okay.
Yes. Mm.
Okay.
Yeah.
Okay, thank you very much. Prof.
Yeah,
Okay.
thank you.
Next question, please. Yes, please. Please come to. Okay. Yes. Thank you, Mas Faris.
Thank you for the opportunity. My name is Dewi. I'm from the doctoral medical science. I would like to ask about the small molecule that bind to the Darcy. In the future, what do you suggest how to deliver the small protein into the Darcy? Thank you.
Yeah. Okay, we screened the small molecule. We found that in vitro system, actually using the PK parasite, because PV parasite we cannot be cultured yet. There is no system to culture. That is why we use a model as a PK. And of course, this small compound is at this stage difficult to use to the human. So we need to optimize more. And also we have to test in the ex vivo culture by Vivax also. Because anyway, Plasmodium knowledgei and Vivax is different species. Even we genomically edited in this PK parasite, anyway, different is a parasite. That's why we need to prove that in the Vivax, it really works or not. Confirm again. Then if they work, yeah, it's still very far from the final goal. Actually, this small compound can be used for treatment agent. At this stage, it's really difficult to say. maybe cannot and now, but possibly in the future, if we make some derivate from this small compound, probably we can develop to the safe or just higher activity of this DPP and DRC interaction blocking agent. The most problem, because recently we used the vaccine that is a protein-based vaccine, right? The protein-based vaccine, we have to think about the stability based on the temperature. Even temperature change, the protein structure will be changed. That means antibody, also quality will be changed. That is most problem. That is why, but small compound is very stable. So we can deliver to everywhere without freezing the vaccine or without make a low temperature, something like this. So this is a good advantage of this small compound used for the treatment, just like a drug, like a chloroquine or primaquine, something like this. But this concept is, as I know, nobody tried in the malaria parasite. So this is first time the study to find the small molecule which can block this essential interactions. So quite challenging, this project. But luckily, we found some of the compounds highly possible to block this DPP and DRC interaction. But the further still remained a lot. So we need to do that work. So maybe someone in here can do that work. Okay, thank you.
Okay, next question, Miss Dewi? No? Not yet? Okay, please.
Okay, thank you for the opportunity. I'm Maria Ulfa from Parasitology Faculty of Medicine. And first of all, I want to ask about malaria zoonosis malaria. I have read a journal about when someone exposed with human malaria, there will be any protecting from Plasmodium knowlesi. And this study from Professor Han want to block DRC binding with Plasmodium 55. That's mean when it's blocked, so the immunity will be not built. Then the second study about the eliminating the malaria from modification in gametogenesis. That same will make the human didn't have immunity from human malaria, mean only block the sporozoid to infected the human.
The human.
Yes.
Okay.
What do you think when its immunity is gone from the human malaria? Will be the Plasmodium knowlesi case increase? Because WHO have reported that the human malaria decrease, but the Plasmodium knowlesi is
Increase. Okay. So your question for Prof. Han?
But
Both of them. Okay. First, maybe Mr. Firman first. Okay. And then Prof. Han.
So the first question about the zoonotic malaria, but I have I mean that the zoonotic malaria, if we expose from the first from the human malaria, for example, Plasmodium vivax or Plasmodium falciparum, I think we have I mean that the immune to protect our body to from infection from Plasmodium knowlesi. But until now, human to human is no evident transmission. I mean that if we found Plasmodium knowlesi patient in the area, in that area, and then the non-infected mosquito take a blood from
The patient?
patient and then transmit to another human, the health human. So and then no evidence.
No evidence yet, yeah?
Yeah, no evidence yet. But from animal to human, yes, it confirmed that. That's mean that until now the Plasmodium cannoli only transmit from animal to human.
Oh, okay.
Yeah, and
Okay.
like that, and then maybe Dr. Han.
Okay, please.
Yeah, about the first question, yes, if we block the invasion of the merozoite, the human immune system will be activated and they will clear it automatically. So merozoite cannot be survived in the human host without any invasion. So our spleen will be clear the merozoite by the human immune response. And the second question is about the vaccine development, right? Is it correct? Second question is about the
Second question is tadi when the human we don't have human immunity from human malaria, then that means that the chance to get Plasmodium cannoli infection will be greater or not?
yes. For example, if I got the Plasmodium falciparum,
Yeah.
I developed my immunity, right? I produced the antibody against the Plasmodium falciparum. After get the Plasmodium vivax, still have Plasmodium falciparum the antibody. So some of the antibody cross-react with the vivax. So we can protect partially the Plasmodium vivax infection. So it quite same, the Plasmodium knowledii and vivax genetically is very similar. They are just like a sister species. So by chance, if I got Plasmodium vivax first and after that Plasmodium cynomolgi got infection, I can protect more easily because I already have an antibody. Of course, this antibody against vivax, but they can cross-react. They also can recognize knowledii and the cynomolgi, even the knowledii also. So we call that is a cross-reactivity of the antibody. So whatever that is zoonotic malaria or not, if the protein structure is similar, they are producing the antibody structure will be similar. They recognizing the epitope will be similar. So we have an immunity for that. So yes, just like that.
Okay. Okay. Next question maybe, Maria?
That means the interaction between the DRC maybe block the Plasmodium cannoli too, because it's same structure.
Yeah, that's true. So today I'm just introduce only one part of my project. Actually, I'm also focused on the protein-protein interaction, as I told you. So we're focusing on the DBP and DRC interaction because DBP has in the many species of Plasmodium, like Plasmodium knowledii also have a DBP, right? Vivax also have a DBP, and cynomolgi also have a DBP. All of they are very similar, but the binding pattern is a little bit different. So in my lab, we express the protein, DBP protein and DRC protein, and we check the binding affinity of each other. And also we're trying to treat in here the antibody to block this interaction. So all of them has a cross-reactivity, especially even the genetically P. vivax and P. cynomolgi more similar. So they have a common effect like that. So that is also one of my projects. And recently I want to expand more because there is a lot of zoonotic species, zoonotic malaria, even the Fildi or Simium brasilianum, there is a lot actually, the Inuai or Simioballe, something like that. All of them have a DBP protein. And human have a DARC as a receptor, right? Even the monkey, there is even the different species of monkey, all they have the DARC. Just amino acid sequence a little bit different. So we want to check this interaction affinity, how much they are different. For that, we can know how this monkey malaria species can infect the human, how much strong the interaction length. So also we can guess the natural host, we can estimate based on the DBP and DRC interaction. This is the most essential interaction. So one of my study, almost done, also that one. Only I'm focusing on PK-DBP, PV-DBP, and PC-DBP. So the result is that the PV-DBP much prefer to select the human. And PK-DBP almost equal. Human and monkey, the binding strength is similar. But PC-DBP is much prefer to select the monkey, not the human. This is one of the results. And also another ligands like TRK proteins or RBP proteins. Also, we're trying to discover the receptor, novel receptor. We don't know, but they can bind with the host cell, but we still don't know the receptor. So we recently found that the PVRBP1A, that is also very important ligand. Now we know what is the host receptor. So this study is really, really important to develop the vaccine because the Plasmodium species differentiate the virus because virus has only one protein cover the seal, we call it capsid, right? So virus invasion is just one ligand and one receptor interaction. Quite simple, just a one-to-one interaction. But malaria parasite is a mammalian cell, like a mammalian cell, right? Eukaryotic cell. So there is a lot of ligand. They have a 10 to 10 pair. And they frequently change. That is why it's really difficult to develop the vaccine. So this ligand, we have to know which receptor interact each other. But in biobox research, even the falciparum same, only very few interactions discovered. So after we can draw whole this map for invasion, maybe we can make a perfect strategy to block the invasion. But still far from that final goal. Still we don't know many things actually. Yes.
Okay, thank you, Prof. Han. Another question? Not yet? Maybe I have one question, Prof. Han. You mentioned about the Plasmodium vivax, okay? What about, is there any other species in Korea, in South Korea? Maybe oval, malariae, and also falciparum. As we know, in our country, Plasmodium falciparum is the most dangerous species. Okay, please.
Yes, the endogenous case in South Korea, even North Korea could be same. Even North Korea, we have only vivax case. Right? The falciparum or other species sometimes have from the imported case from the outside. But from the nearby the airport, they already detected the parasite. So we can quickly treat the patient. So there is almost no chance to spread other species in Korea. And also in Korea is very interesting country. At the winter, it's very cold. Whole nation is freeze.
Yeah.
So that means mosquito cannot be survived. They cannot be activate in the winter season. But why annually it occur the vivax patient? So maybe many people just say like that in Korea vivax character is a little bit different. They have a very long haplozoid stage. So sometimes they just stay in the liver several years, and they just pop up when the human and the host immunity decrease, something like this, or any suitable condition occur, they just pop up from the liver. That is why every year there has the malaria patient, even there is winter season. Right.
Okay, and I think, and one more. what about the factor? What the mosquito? What the species that you find in your country? Only Anopheles or maybe other species?
Yeah,
For
the
this?
major vector for the malaria in Korea, yeah, Anopheles, definitely Anopheles.
Definitely.
And nearby the DMZ region, just in case DC, they told us maybe the, what is that, Anopheles sinensis is a major vector in nearby the DMZ. But when we study about them, the major vector study, maybe not the sinensis has another species like a Kleini. The kind of one is could be the major species. And from this year, not from this year, several years ago, the malaria patient previously just occurred in the DMZ area, nearby the DMZ. But now it going down. And even the southern part of the Seoul also have a patient. Oh. Because the sinensis majorly live in the nearby the DMZ, but Klein is the southern part, they live in there. So recently we think about that maybe the Klein is the major vector of the Korea malaria.
Okay. Yes. Thank you. Thank you very much, Prof. Han. Okay. No questions? Okay then. I think at the conclusion of this guest lecture and also discussion, the first diagnostic, especially molecular diagnostic, is still challenging in our country, Mr. Firman. And also there is cross-reactivity antibody between species in Plasmodium. Okay, applause for the both speaker. Thank you very much. And now I give the time to MC. Please, Ms. Sinta.
So thank you very much for both of the lecturer, also the moderator for Dr. Ada. And now, please stay seated at the stage because we have an award plaque as a token of appreciation from Faculty of Medicine, Universitas Indonesia. And I would like to invite Professor Wawai Muli as head of master's program in biomedical science, FMUI, to give the plaque first to Dr. Jin Yi Han. I'm so sorry. And the second plaque will be awarded to Dr. Nurhadi Eko Firmansyah. Thank you very much, Prof. Wai. And the memories will linger forever. So now, as our custom, we would like to take pictures for both of the lecturer, the moderator, and also Prof. Wai, the documentation. And I would also like to invite the partner from KNU to the stage for the photo session. I would also like to invite Ibu Rawina from Malaria Research from the Department of Parasitology of MUI. And we would like to invite everyone to come to the stage and in front for the whole audience photo session. I mean the offline audience photo session. For all audience, please come to the stage for the photo session. Okay, thank you very much for the audience. And once again, let's give a big round of applause to Dr. Jin Hee Han from KNU and our own Dr. Firman Shah from FMUI. And this event is made possible by the collaboration of Master's and Doctoral Program in Medical Sciences, Faculty of Medicine, Universitas Indonesia. My name is Sinta. See you in another lecture. Good morning.